Hello, I'm Meredith Blackwell, and I'm
Hello.
here with David McLaughlin,
Hello.
and we are at the International Mycological Congress 11 at San Juan, Puerto Rico. And David is a professor emeritus
Meredith.
at the University of Minnesota, and so we're going to talk to him a little bit about his mycological career, which has been really good.
And curator emeritus.
Curator emeritus. Sorry.
Two.
That's important. Okay, so where did you grow up?
I grew up in Rhode Island, in Pawtucket, and a somewhat strange upbringing in that I lost my mother very early on, and but I was very influenced by them, both my parents. They had a very strong interest in education. Neither of them had gone to college. My father didn't graduate from high school, had to go to work to help support the family. But my mother did graduate from high school. But as kids, we had this very interesting thing. When I grew up, I realized we all had desks. We had a very strong interest in the value of education. So we were essentially being programmed.
Yeah.
So.
Yeah, my dad's family was that way with third grade father and a sixth grade mother, I think.
Wow.
Yeah.
Wow. Right.
Yeah, but luckily I got the next generation.
Yeah. But they were very interested in education, and I think they read, they did all those kinds of things that really they were involved, even though they didn't go to college.
So in high school, were you interested in fungi? Or?
No, not really. What I was interested in was plants. I found that I like to work with plants. I like to grow them, do things with them. And so I decided I would go to college and go to work with plants. But what got me interested in fungi, I went to Brown, which is a small botany department, very much knew all the professors, wonderful environment. They hired a new faculty member just as I came there, Mel Fuller.
Oh.
So
I never knew Mel had been at Brown.
Yeah, he was at Brown for about 5 years. He was there all the years I was there. And I got interested in fungi, though I started working on the side, just helping do projects and things. And I guess right at the beginning, I worked with George Church, who worked with grasses, helped with hybridizing. But when I eventually ended up working with Mel and isolating marine chytrids. So
hard thing to do.
Oh yeah, seaweeds and special augurs and things like that. But I always had a real hankering to work with larger fungi. And my last year, I was actually able to work with Wally Snell, who had been retired, and his
He's
assistant.
a former baseball player.
He's a former baseball player, one of those rare breeds.
Was it St. Louis Browns, or I can't remember now.
I think the Red Sox, but
Red
I'm not
Sox?
positive.
Okay.
Yes, and he always talked about having been a catcher for Babe Ruth.
Wow.
He also was full of stories, so.
Baseball stories.
Oh yeah. Well, all kinds of stories.
Oh,
no, he was actually also a coach for when he was still a faculty member. He was one of the coaches for the college. Yeah, so he was always interested in sports. I think he played 3 sports in college.
And before you go on, I just want Snell and Dick.
Snell
was
and
Dick
Dick
there when you were there?
Yes, she was the departmental secretary, and after his wife died, he married her. So it eventually became Esther Snell.
But the book that they did, was that before they were married?
I think it's Snell and Dick. Yeah, yes, I think it was.
Yeah.
And that was in production for a long time. I mean, it was all his watercolor paintings.
Wow.
There was no way to capture the colors of the boletes when he was growing up. So he taught himself how to do watercolors, and the rest is history.
Okay.
Yeah, but I got to travel with them too. I went first with Esther Snell. We traveled to Canada and up into Maine, collecting fungi in New Hampshire. Came back out of the woods one day. We stopped at a brief trip and came out with this beautiful red fungus. Boletus flamans, never seen before.
You're kidding.
No.
Bright red. And
bright red and never
it.
reported.
Did it not fruit very often? Or?
All I know is that we found it under the ferns in a really beautiful place in New Hampshire. And you had to kind of look under things to find it, but I think it was with hemlocks. So
interesting.
yeah. So
So then from Brown?
From Brown, I actually went, after I graduated from Brown, I went to McGill for a year and worked with Charles Wilson. While I was at Brown, they encouraged me to do what I wanted to do, so there was great interest in growing and fruiting mushrooms in those days. And there were a few reports of boletes in culture, so I started isolating boletes, and they said, go ahead and do that. By the time I got done, I had one that fruited in culture. So that I started working on that more. It fruited when I was in Canada, and when I came back to around the next year, Canada was not a good situation. The university had problems, underfunding. It was political in part, the underfunding. Tensions between an English university and a French government system, and so I came back to Brown. Mel was still there. He was about to go to California. I went to California with him.
I thought there had been Mel more than once in your life.
He was, and so I went through graduate school with him. So and I worked on studying that bolete, the environmental control of fruiting in that bolete. And
wow.
So.
Yeah. So then, after you got your degree, what did you do?
I had a postdoc to Denmark, to the Genetics Institute, and they were very interested in synaptonemal complexes and the structure of synaptonemal complex. And so I ended up working on doing nuclear division in Boletus rubinellus or now Chalciporus rubinellus. So yeah, so. Yeah. And then there was all great interest in cell structure in those days, and so I continued working on that. I came back under the position at University of Minnesota. I had actually applied for it and knew I was going to go there before I went on the postdoc. They wouldn't let me stay a second year. They insisted that I come. So.
The synaptonemal complexes, it reminds me of my PhD qualifying exam. Harold Bold, who was in the department at Texas, asked me, that was the first question asked on my exam, and I didn't give him what he thought was the correct answer. He thought it was the chromosomal structure itself instead of the stuff that holds it together. Is that correct?
Yes, it's a mating structure when they come together. In fact, that was one that
protein.
It's proteins, I think. And what was really interesting. Mogens Vestergaard at the Genetics Institute had worked with a particular ascomycete, names from escaping me, that had unusually large chromosomes. And when they did it, they had a very complex structure in the chromosome with the mating, so they could see a very detailed aspects of the mating, and it controls crossing over in the
well, he thought it was the chromosomes itself.
And
the upshot was that the next 10 minutes they argued about it, and Howard Arnott was on my committee, and he eventually is an electron microscopist.
Yeah, so straightened it out.
It was funny. You reminded me of that.
Yeah.
So you were lucky then. You had a job to come to.
I had a job to come to. In those days, there was a real interest in hiring people, so there must have been a lot of openings. Actually, I think it was also the time of Sputnik, and they were interested in keeping up with and getting into the space race. In fact, as a graduate student, I had a space NASA fellowship at one time, and I was studying mushrooms. Well, then maybe there's some on Mars, right?
What year were you hired?
I was hired in 69 is when I started.
Yeah, interesting.
Yeah.
So did you have to build a lab, or was there something there already?
No, there wasn't much. There was a little bit because somebody had left, a little bit of equipment, but basically I had to get equipment, set it all up, and so forth.
And you were a curator, as you pointed out.
Well, but I wasn't hired to be a curator.
Oh, you weren't?
No, that came later. That came out of interest.
Okay.
Yeah, and the fact that I had gotten to know somebody in Minnesota, Margaret Weaver, Peg Weaver, who was interested in fungi. She was so-called amateur, but a very gifted amateur, and very, very professional. And we had gotten some collections in, I think, from her, because she was getting on in years. But nobody was doing anything with it, So I eventually kind of got in there, started to get things organized. We had a fungal curator in theory, but he was only interested in the lichens. It didn't go beyond that.
So you had
yeah.
a basis of a collection in was it in the Bell Museum?
It was not. It was in the botany department at first, which then eventually became plant biology. But it was in the department. It only later did they decide to go in with the other curator and move it under the auspices of the Bell.
Okay.
Yeah. So. But it's a huge collection.
Yeah, so who had started it? There had been other mycologists there before you besides
Oh yes,
the lichenology.
Clements was there around the beginning of the 20th century. And some of the other people in the department had some appreciation for. I'm trying to think who else was mycological. There were. I'm thinking. I can't pull up other than Clements right at the moment. But some of the other people in the department would not pass up something like that. We have tuber lyonii, which was collected by one of the plant taxonomists in Minnesota.
Yeah.
So.
And then there's a rust lab now, a USDA lab. Was that there at the start, or is that later?
It was there when I came to Minnesota. I was there for quite a long time because it went back with Stakeman, I think.
Okay.
And but the collections were in two different places. There was some in Minneapolis where we first were, and then they had split the collections in the 20s, and the other part was sent to St. Paul because the plant pathologists needed access to some of that stuff.
Yeah.
So eventually, I got together with somebody, and we put the two collections back together again. The plant pathologists were vitally interested in taking care of them, and we were. So it's actually a fabulous collection. We have all the type specimens for many of the rusts of Central and South America. And it was collected by a banker who retired, and his hobby was climbing mountains and collecting rust.
What was his name? Do you remember?
Yes, Hallway.
Hallway, okay.
Hallway, yeah.
Yeah, that's a second banker I know of. There was another banker at Harvard that did the same thing.
Yeah, and he worked with people like Arthur and at Purdue, and so and other people like Jackson in Canada. So all the specimens were, very professionally handled and published. So.
Well, I guess one time we had did we have a joint paper with the Terriula-like thing that came from ants, or was that your student?
No, we did.
Yeah, I think we were both authors.
I think we're both authors on that.
Yeah.
And that was, do you want me to talk about Terriula?
Yes, I do.
Terriula. The Terriula story is kind of strange. I was in graduate school. They had brought Rudy Reichly had asked someone to bring him back from Trinidad some cocoa twigs that were infected with a basidiomyces, marasmus something or other, it's escaping me, but he had them incubating in the lab, and up comes this fungus on the twig. So he says to me, that's probably a basidiomyces, you could probably culture that. He had some carrot agar. I picked a fruit body off and planted it. The rest is history. It grew, it fruited in culture. We kept it going, and we did stuff with it, and and eventually we published it, published a new species, and the name is good, Terriula echo, because the culture reflects the fruit body. It does the same thing on the surface that it does on the fruit body. So it's quite neat. It's not common to do that in Terriulas as we have in culture. So we pursued it. We began, even though we were working on some other project, we continued to work with that. And finally, Bryn Denninger came to the lab, and we got some funding to help me look at some of the evolution of the clavarioid fungi, and he included that in it. And that began the process of really understanding it. And I think it's about the time that you sent us the culture from
a student isolated it in
as
class.
An epiphyte.
Yeah, from Magnolia, I believe.
Right.
And then I also had come across it over near a plantation where actually Bernard Loewy's ashes are scattered.
Oh, really?
Yeah, a state state property there
Huh.
in Louisiana.
Huh.
Yeah, and I found it there on leaves.
Right, but nobody knew that it was an epiphyte. We saw it, I think, after that, maybe just before we came to Baton Rouge, when we first came there for Thanksgiving, from collecting in Puerto Rico. We're out with Gene Lodge. Here's a leaf caught on a tree, still green, and here's a tariola coming out of the leaf.
Proof
In the field.
positive.
Well, and then Ron Peterson referred somebody from India who thought he had a parasite on palms to us to look at. That's another tariola. And then the other thing that happened, I think, with the molecular data that Bryn was working with, we found out it was one of the missing groups of the fungi being cultivated by ants.
Right.
So
The one that wasn't a real mushroom.
right,
Yeah.
right,
Yeah,
exactly.
yeah.
And so it turns out it has all these different roles. Everybody thought it was just a saprophyte. It's anything
Yeah.
but. It's
Very
everything.
interesting. Yeah, well, you've worked on a lot of basidiomycetes
Yeah.
that no one ever heard of.
Yeah, well,
And-
I love diversity.
do.
That's one of the things.
But you've also worked on
captured me.
ascomycetes, and,
Yeah.
and your micrographs are beautiful.
Thank you, but
Yeah.
it also helps with, the really good students who
Right,
have done the work
right.
too. I think the students have been fantastic.
You've had some good students, and you've also had a lot of, well, you've helped along with the molecular phylogenies by finding characters that correlated with it.
Yes, we did,
Yeah.
and actually one of the most interesting ones that you can plot them out. You can plot out the nuclear division characters, and you can see patterns of evolution in the characters. One of the ones that turned up recently, we looked at Ariscalpia, which is a hydnoid kind of thing
Yeah.
with teeth. And also, we know it's in the Russulales, so we looked at a Russula, and it has a strange vesicle. It's though you push your fingers into the vesicle. It's like and it seems to be unique to the order. So that's one of these cases where we may have an organelle. It may be the organelle that's responsible for accumulating the sticky substances, like the lactarius have. Yeah, but all these other fungi have equivalent sorts of things.
Yeah, interesting.
So
That's not published yet, huh?
Oh, that is published.
Oh, it is?
Yeah,
Oh, I've missed it.
Yeah, and thanks to a student now working on a PhD from Brazil in Wales, we had got him involved in interest. He's turned out to be great.
I think I met him the other day.
Yes, you did, Kale.
Yeah.
And he had figured out one of the things that was in the Terralaceae actually belongs in the Russulales. And it's got
Amazing.
some of these kind of glial hyphae.
Amazing. Yeah.
So there's some neat characters
Yeah.
there. So
And then I like the work that you did with Tharbelia and comparing it to the rest of the so-called discomycetes.
which one?
It was
Orbelia. Yes,
Orbelia.
yes. Yeah,
and showing that there were differences between it and the other.
Right, the
things.
Pizizales, yeah, and the characters sort of fit that is basal in the group. They're relatively simple. And then Roseanne looked at the septa in Neolecta. It's down
Right.
with the yeast groups,
Right.
and the way the septa get blocked is unlike anything with the Pizizes where you have the Waronin body. It uses a vacuole with a crystal.
Yeah.
But everybody has thought that was a very independent lineage among this basal group. Seems to be true. Yeah. So. Yeah.
So it's really been some great work.
Yeah.
And I should mention you have a helpmate, Esther.
Oh, Esther's been great. She's always supportive. It's nothing like having two botanists together.
Yeah, I know.
And it's having somebody who'll help with the photography and likes to go out on field trips with you.
Yeah.
So it's been great.
Yeah.
We've continued, since we retired, we've been continuing
You've
to
been doing a lot.
get out there.
And you've got money.
Yep.
And I want you to come back to the Congaree Swamp.
All right, we'll try to do that.
Our poorly known national park
Yeah.
in South Carolina.
Yeah, well, Minnesota is so badly known, and I'm still
know.
working on it.
I
I'm
know.
hesitant to get myself into too many things, but I think we'll come back.
Yeah.
We will come back. Yes.
Okay.
Yes.
Well, thank you. It's been really great to talk to you.
It has been really fun.
Yeah.
Yeah.